Um, okay, here we are. Now it's my pleasure, very pleasure, to introduce the next speaker. It's a featured keynote that we, as ARM, are very happy to host today. It's a scientist that has been really changing this space and pioneer in this space, especially in the cell therapy space. Professor Massimo Dominici, please join me on the podium, and please join me in welcoming him.
So thank you very much for the introduction and for the invitation. I'm Italian, as you understand, and I'm honored to be here, somehow representing the country and all the excellence that we have been seeing in this first day of the meeting. And essentially, what I would like to share with you this afternoon is some experience that I have been done either at the academic level or at the industry, and just trying to share the challenges of developing cell and gene therapy, but also looking at the opportunities that are appearing. These are some disclosure. I was founding a company that was sold to another one, which is called Evotech, and I'm recently founded a company called here Biotherapies, and I will be speaking about that. These are the messages that I would like to share with you. So we'll start with the 3 examples of cell and gene therapy developed at the academic level, at the SME level, and then a CAR-T strategy that is still in academia looking for SME venture. And then we'll be asking to ourselves why the cell and gene therapy seems not to be delivering as we expect. Is this normal, or is this part of a physiological development? And then how we can fill these gaps for making these things real go to the clinic, as we have heard from the patient just a few minutes ago. So I'm based in Modena, not far away from here, from Rome. It's a small town which is very famous for 2 things, actually. One is balsamic vinegar, which I'm sure you know. And the other one is sports cars, because we have Ferrari, we have Lamborghini, and we have Maserati. And I'll be speaking about car, not car, maybe CAR-T later on. So you'll be apologizing for that. But we'll also be speaking about fine manufacturing and GMP manufacturing, I would say. Why I'm here this afternoon? Because I believe in cells. I believe in cells as a vehicle to treat patients with incurable diseases. And in what we are trying to do in Modena, we're trying to use MSCs, CAR-T, and biomaterials together, trying to fix 2 major aspects: regenerate tissues, mostly for bone diseases, but also to treat cancer. I'm an oncologist, so it's a clinical priority for me, but it's also a scientific passion and a deep interest. And we use 2 different cell types. One is mesenchymal stromal cells. We isolate them from different sources, from bone marrow particularly, but also from adipose tissue. And for bone marrow, we use them for bone regeneration. For adipose, we use to treat cancer. And examples, these are European public-funded clinical trials when we use MSCs for the treatment of bone diseases in humans. It's an autologous process, a fresh product from bone marrow down to the patient using biomaterials to combine into a combined approach of TMPs. This is just an example. In the upper part, the syringe is containing freshly isolated MSCs, and in the lower syringe, you see the biomaterials. They're combined, and they are treated, and they are implanted into the bone of a patient with a variety of bone diseases. We treated Joe diseases, non-union defect, and osteonecrosis, but you understand I'm not an orthopedics, and my major interest at that time is to focus on MSC function and a way to predict bone regeneration. Essentially, we realized that when we were treating patients with MSCs, there were some MSCs which were very good making bone. Oops. Something was happening. Nothing major. I just want to assure you. We'll use MSCs just in case to regenerate the podium here. So for the bone regeneration, essentially, we realized that in some cases there were patients which were healing, in some patients which were not healing. And then we ask why MSCs, which are notoriously making good bone, why in this patient were not performing. And so we address by a complex in vivo, genomic, and simple in vitro assay if there is a way to address this heterogeneity and to understand if there are genes which are highly expressed in the most performing MSCs. Out of this investigation, we realized there was a panel of few genes which were expressed in the highly making bone MSCs. And so based on these studies, we identify these genes and we are using them to predict the function of MSCs within either autologous setting, but mostly in an allogeneic setting. So essentially, we were able to get money from European Union to implement a first large multicentric allogeneic MSC strategy where the cells will be selected based on a potency assay, which is based on genes in vivo assay. In this way, we want to be sure that the cells will be implanting, will be performing for a variety of citizens which were affected by bone disease. So what we have been learning from this first experience performed at the academic level in a multicentric way. That there are regulatory challenges within a multicentric study. This is not news, but it's feasible to use the same IMPD in different countries to produce the same product coming from an autologous source, which is certainly challenging, which challenging in terms of logistics to delivering the raw materials, challenging in sending the MSCs from the GMP facility into the surgical rooms, and challenging in terms of combining the cells with the biomaterials for therapeutical purposes. Something which is, I think, sometimes largely underestimated, that the final user is not the scientist that conceived the product. These are surgeons. They have limited time to perform the treatment, and they might be skilled to do that. So the training in the academic center for the final user is so important so that we don't jeopardize a very precious product. Of course, my interest is to understand the performance of MSCs, and so essentially what we are doing now, we are combining prospectively the genes which will be highly expressed in the potentially MSCs making bone with the clinical outcome. So I'm moving to the second example. We're using MSCs to treat cancer. MSCs are tough cells. And they can be modified, they can be loaded with drug, they can be loaded with oncolytic viruses. And after that, they can deliver the payload successfully to the cancer environment. And we are doing that in a variety of settings, in sarcomas, in pancreatic cancer, in brain cancer, and in a specific cancer type of lungs, which is called mesothelioma. These are generally highly neglected diseases where we have 1 or 2 lines of chemotherapy. So it's the perfect, I would say, unfortunately, scenario where we can challenge ATMPs and cell and gene therapy. And this is the technology that the biotech company that we just founded. I'm sure that you're aware of the gravity of pancreatic cancer, which is our major target here. It's a cancer that is growing. It's a cancer whose prognosis is unchanged in 20 years. It's one of the few cancers that hasn't been touched by immunity, by small molecules. And that is an incredible limitation. In this country, there are 15,000 new cases every year, and the survival, it's about 6-8 months. So we thought that this would be the first target, and what we did, essentially, we created a product which is called RR001, which is MSC's delivering a very potent anti-cancer agent, which is called TRAIL. This is an autologous product that we design to be implanted intratumorally into locally advanced pancreatic cancer, using adipose cells as a vehicle and validating this technology, considering that pancreatic cancer generally has a lot of TRAIL receptors. The strategy is very similar to the one that I was showing you in the context of bone regeneration. Autologous cells, in this case, we gene modify them with a retro lentiviral vector encoding for a soluble variant of TRAIL, and then we're putting back into the patient. TRAIL is a very potent molecule which was used as a recombinant factors without big success, but using the cells as a vehicle to deliver the product successfully into the patient. We wanted to combine the capacity of the cells to engraft into the tumor, and then their capacity to release a transgene and then a protein. So the mechanism that we are relying on is we gene modify the cells, we implant them back into the patient, into the pancreatic cancer, and also we give the patient at the same time chemotherapy plus gene therapy. So and why this concept? Because we know pancreatic cancer is not waiting for us, and I think we need to consider ETMPs and cell and gene therapy as part of many different molecules that can be applied to the patient. I think we have been given sometimes too much responsibility to a single cell and gene therapy product, forgetting, at least in cancer, that so far polychemotherapy, many drugs given together has been very effective. So I think we need to get into this mood also for cell and gene therapy, without minimizing their impact or their function, but just considering the reality of a cancer, of cancer cells, and their polyclonality. So this is the strategy that we are doing. The study has been approved by our regulatory agency, so we'll be starting this year, and we'll be combining an early systemic treatment with effective local controls and hopefully with a faster surgical removal. And this is part of a study that we started in the academia and then moved into an SME concept. At the same time, in my lab, we have been developing a CAR-T for solid tumor. And of course, it's not a Ferrari, but we hope that we'll be performing as a Ferrari, as you will see here in the slides. So which type of targets are we challenging? We have an anti-GD2 CAR-T that we are using for preclinical treatment of glioblastoma, of malignant melanoma, and of small cell lung cancer. And we isolated our strategies to work on primary cells, on autologous lymphocytes, to validate preclinically, in vitro, and then in vivo if the strategy is actually working. Just sharing some data with you. This is the first approach that we published a few years ago against the glioblastoma. Essentially, we isolated primary cells from patients, and at the same time, we isolate the lymphocytes, we gene modify them, and then we start to co-culture. So the tumor cells have been modified with the red colors and the lymphocytes with the green as a GFP. This is just an example of simple in vitro approach. You can see in the first row tumor cells from a patient which were growing, so they get to confluence. But when we use allogeneic GFP cells alone, you see that there is no big impact, the second and the third row. When we load the lymphocyte with the CAR-T, you see a lot of lymphocyte clusters, and the red color is essentially disappearing. We moved this technology into in vivo animal model using an orthotopic model, and we wanted to do less as the hematologists like to do. So you have the cancer, then you take lymphocytes, you put the lymphocyte in vein, and then you hope that the lymphocytes are reaching into the tumor sites. Unfortunately, for solid tumor, it's not the case. And so we challenged the approach, but there was no difference. And so intravenous approach is not always good when we want to target a solid tumor. These are examples. But when we change the strategy and we implant the lymphocytes intracerebrally, we realized that the impact was evident. And so we were very happy that we were able to demonstrate some functionality. And as I was telling you, we're moving to an autologous platform. This is just an example, just published a month ago. In this case, we are being isolating lymphocytes from the same patient with glioblastoma, tumor cells from the same patient. We mimic essentially what is actually taking place for the autologous lymphocytes in hematology for leukemia and for lymphoma. The same concept. The bad guys are red, the good guys are green. As you can see here, the same concept. This is a cell line from patient. You see when we mount the CAR T into the lymphocytes, they start to target cancer. This is more evident when we created the spheroids. Essentially, we created spheroids of glioblastoma, and we target them with lymphocytes. And as you can see in the right column, the use of autologous lymphocyte was associated with a massive anti-cancer response, considering that these patients are generally heavily treated and they are taking steroids for a long time, and even sometimes chemotherapy at the same time where we harvest the lymphocytes. So it's possible we can generate this type of strategy. So while we developed this strategy in the lab, I founded a company which is not far away from Modena, which is a cell and gene therapy facility with classifier environment, and with the concept of transferring these discoveries into a clinical reality. Now is owned by Evotec, and we are collaborating with them to develop our cancer gene therapy product for pancreatic cancer. So what we have been learning when we were moving a cell and gene therapy from academia to SME and now into the clinic. First, suggestion to interact with the regulatory agency as soon as you have something good in your hands. Even if it's not perfect, doesn't matter. Go to them, share the strategy. It's so precious because they can help. You can develop the product together with regulators. Either it would be EMA, IFA, or FDA. We had a good experience. We had also good experience in accelerating R&D validation using 3D models to mimic complex cancer like pancreatic cancer, which is very complex to be mimicked in vivo. But at the same time, we've been facing the challenges of transferring all the academical product into GMP manufacturing. Because, of course, the protocols that you have in the lab sometimes not really apply to a GMP compliance framework. So that was very challenging. But at the same time, with the support of consultant, we were able to do that. We are stuck on autologous product in this phase, but sometimes it's mandatory, particularly for CAR T, for the current state of our development. The big issue of viral vectors, because we have been using viral vectors in the lab and then we move into a GMP facility, so we have to change the technology from retro to lenti. In this case, proper partnership can make a big difference. And then there are a couple of other aspects which are not trivial. Transferring academical scientists into a GMP environment is not easy. It needs to be a mental change, which sometimes is easy, sometimes is not really easy. So I recommend to take care to this. And the fundraising has been an issue, but has been always something that we were able to fix if you have a clear target and a clear strategy in the time. So coming from all these two experiences, the last part of my presentation is asking to ourselves, why this is so difficult to make this cell and gene therapy with market approval. Because at the end of the story, we have hundreds of MSc trials in clinicaltrial.gov, 1,000 more than 1,000 CAR T trials, but a few of them are reaching the clinical prime time and the market authorization. So to answer this first question, I was looking into the past. I was looking at the monoclonal antibody. So this is the first example of the monoclonal antibody, which was marketed in 1986 it was called OKE-T3. I'm sure you're aware of this old technology. It was an anti-CD3, was used to generate immunosuppression. But the first scenario of this clinical approved antibodies, that there were side effects. There was a generation of mouse anti-human antibody. And also there was issue on the hybridoma that was producing the cells. They were genetically unstable, something that reminds me new technology related, for instance, to iPS. But still, the product was approved. And then I was asking, sometimes when I look for answer, I use the chat GTP. And in this case, I was asking to chat which have been the major challenges in the early monoclonal antibody development. And I was surprised to find the same challenges that cell and gene therapy are facing here. You know, technological limitation, specificity, and immunogenicity, production scalability, limited targets, and regulatory hurdles and costs. So the same. These were, not that long ago, but still asking to the artificial intelligence. The answer was that despite these challenges, we know that FDA every year is approving several 30, 40 new monoclonal antibody. So we hope that this sentence will be good also in the next few years for cell and gene therapy, accounting the same challenges, which were at the end fixed by scientists and regulators. So which are the current challenges for cell and gene therapy development? Well, let's start to consider that, as in my case, most of the cell and gene therapy technology have been developed by small and medium-sized enterprises. That these are complex products, which challenges related to product quality, scale up, clinical programs, and cost, and reimbursability. And then I was trying to dissect which are the major obstacles to bringing this product into a clinic in a solid way. So let's say that I was dissecting 4 major obstacles. One is cell-related obstacles, because these are living cells, process and scale-up related obstacles, and then what I call the original sins of academia. I'm coming from them, so I can say this. And then there is the issue of cost and reimbursability. So very briefly, each of them. Cell-related obstacles. We are not dealing with pills. We are dealing with living cells, and each of us has a different MSC type, as I was demonstrating. My MSCs are making bad bone, Miguel MSCs are making super bone. So why is that? We need to address this aspect either into an autologous setting, but what if my cells would be the source of an allogeneic product? It's going to be a disaster for the product, while MSCs for Miguel would be super. So, and we need to understand that. We need to take this into account when we are designing clinical studies. But there are also process and scale-up related obstacles that are related to a variety of aspects which are summarized here. Well, most of the cells that we are moving into GMP facility have been manufactured into a lab with the limitation of reagents which are not GMP grade, protocols which are not GMP grade, aspect related to SOP which are not even minimally GMP grades. So moving all this technology into a GMP environment generate obstacles that we need to overcome to make this product reach the clinical prime time as we all want. And then the examples in our case, but then curiously, 2 other groups published a month ago, the way in which you are delivering the cells can make a difference. Two different groups targeting glioblastoma with two different CAR T. The first was able to get some result because they were able to inject the cells intratumorally. The second group, they were not able to get the result because they were trying to reach the tumor after intravenous injection. So the delivery way is not that one fits all. Each tumor type has to be managed in the right way to reach the target in a consistent and profitable manner. And then I'm going to the original sin of academia. Well, as I was telling, most of the products are coming from hospital and from small labs, which are carrying the bags of early academic preclinical study. Aspect related to limited space management team and sustainability may limit greatly the implementation of and the transition of this product into the clinic. Regulatory aspect related to early phase may hammer regulatory, an academical based lab, which might not be equipped to face this aspect. Sometimes there are lack of dedicated grants, and this is also a limitation generating obstacles. And mostly what I think is important that the partnership between those labs and the more experienced CDMO or CRO is taking place very late during the development. And this is something which is limiting greatly the development of the product. So how can we, I'm almost done, so please apologize, but I wanted to share my vision on this. So how can we fill the gaps and get this funnel bigger to make more product? Well, first of all, being aware of these obstacles is an opportunity. We need to facilitate and accelerate phase 1 study by novel regulatory approach. The FDA is an example. Here in Europe is sometimes very difficult. We need to have a better integration, as I was saying, between the CDMO and the pharmaceutical industry, starting from the SME. Favor non-profit national authorities that may facilitate the link. We have examples, Catapult, CCRM, and others. And then learning from mistakes as we have been doing and sharing. And lastly, the innovation. I think the innovation will be a big change in the next generation cell and gene therapy. When I mean innovation, I mean the advent of a new vector to modify our product, new processes related to media optimization and rapid expansion to minimize the ex vivo expansion time. But also in the introduction of automation and integration between different processing parts. And this will take some time. This is some expected time that are not so far away in the time. So I think it's very important to consider this aspect. Other two innovation aspects which are very important, digital innovation, which is entering in many fields and has to be entered also into a GMP facility. In a different ways, from electronic batch record to real-time monitor. And last, the point-of-care manufacturing, which would bring in a more diffuse way the use of cell and gene therapy very close to patient, which has limitation, but is a reality that is growing. If we're able to face this innovation, we'll be able to reduce the cost of manufacturing. This is a very nice paper that I recommend, where they were dissecting the introduction of each innovation is going to decrease the cost of manufacturing, because this is what we want. We want to innovate, and at the end, we want to have a product which is less expensive. Innovation will also drive from autologous product, which is what we have now. It's like airplanes. We have airplanes. We're using them. It is a cost, as we have 6 CAR T products approved into the market, which are a pain, but this is what it is now. But the development of the technology is bringing us to the moon somehow, to have an allogeneic cell and gene therapy as a universal, viable, ready-to-use cells that can be stored and can be used as monoclonal antibody. But also we have been seeing in vivo cell and gene therapy, where we can dig into the ocean of let's say, of the patient, in this case, and modify the cells inside with the limitation that we are actually currently developing. There are some positive signs in the field, yes. This is the latest report that I was downloading on the web, is available. And you see the number of, the percentage of product that the FDA approved in the cell and gene therapy increased in the last year, but also the trend of predicted FDA approval in the next 10 years is substantial. So there are very positive signs under this point of view. So I'm ending here, thanking you for the attention, but I wanted to remind something that someone very famous said related to the moon mission of the NASA, that they choose to go to the moon in that decade, because it was a few years ago, and do other things. Actually, we are the one they want to do the other things. So thank you very much.